Hello students, are we live? Let's see, 1 second. Yes, we are live. Is it? Let me just see the screen if everything is visible. Yes, we are live. Yes, we are live. Okay, so hello students, I'm sorry the class got a little delayed. I was caught up with some other work. So today we are going to start molecular basis of inheritance. So I don't know how many of the students know the importance of this chapter. This chapter has a weightage of around 8 to 9 questions. Okay, how much? 8 to 9 questions from this chapter. That means you will get 36 to 40 marks from this chapter. Okay, so it is not a simple chapter. It is a chapter that you have to take care a lot. You have to give a lot of importance to this chapter. Okay, so students, today we'll be dividing this chapter into parts because the chapter is very long. It will take around 7 hours if I do a marathon. So that's why we will split the class and we will do it in parts. So today what we are going to focus on is we are going to understand the basics of this chapter. Okay, so I will not be complicating anything. I will try to make it as easy as possible. Okay, my voice is clear. Try to make it as easy. Yes, everything is clear. So yes, do share this link with all your friends who are preparing for boards because this chapter is very important from board point of view as well as from your NEET point of view. NEET point of view, this chapter is ultimately, this is like a, this is like a one-shot go chapter. And you see at the end of the session, this chapter will be so easy for all of you. Okay, so shall we start? I know, okay. So yes students, the chats are off, it's okay, it is just to keep you more focused on the class. So please write down notes with me, and if you want this notes of all these chapters, please do follow us on our Vedantu NEET English Telegram channel. And those students who are feeling that, in this channel now a lot of classes are not happening. So in case you feel very, demotivated and you feel like, how do I prepare for NEET? I do not want to join any courses, I want to do it for free. Then please do join our channel, that is Vedantu NEET English. Every single day long marathons will be happening. We will also be doing PYQs for each of the chapters. So it is like a one-stop solution. You will get everything. You will get the chapter detailed lectures, you will get PYQs, you will also have mock test with us. Okay, so quickly join our channel and do not worry. Students, NEET is more important, not the channel. NEET is more important. Shall we start? Okay, quickly share the link with your friends. Okay, and we are going to start. Okay, so students, when we talk about molecular basis of inheritance, the basic thing in this chapter is DNA, okay, gets converted or gets, changed to mRNA. mRNA gets changed, okay, to your proteins. Okay, proteins. Now, the thing is that this entire chapter is only focusing on this. Now, when DNA forms its own thing, it will be called as replication. God, why is this not writing? Since 1 second. Okay, replication. Is it writing properly? It's not writing, no? Okay. Yeah. So when DNA forms its own, its own copy, it is called replication. And when DNA gets converted to mRNA, it is called translation. Yes or no? Translation. Okay. So students, yeah, this pen has some problem. One second. Yes. Translation. Now understand. Yeah. So understand that when DNA gets converted to mRNA, the process is called translation. Now mRNA gets converted to protein. It's called as transcript. Sorry, I wrote it ulta, no? 1 second. There's some technical issue with this. Okay. Yes, sorry. First is transcription. So give me a minute. I'll just fix this. I will just fix this. Can you see? Yes. I'm not able to write it on the PowerPoint. That's the reason. Let's try this way. Yes, students, now I think the problem will be fixed. Do not worry. But the screen is white. Okay, but the screen is white. Is it okay? Yes, fine. Okay. Give me 1 minute. Yes, fine. Okay, students, so what I was telling is, now the technical issue is fixed. So, we will continue the class. Yes. So, when I was talking about this, understand that I was trying to say DNA. See, now I can write smoothly, right? DNA will get converted to mRNA. This mRNA will get converted to protein. This is the whole chapter. To be frank, only this much is there in this chapter. But when it comes to this, you have to understand a lot. So when DNA makes its own copy, it will be called as replication. Okay? And when DNA gets converted to mRNA, it will be called transcription. And when mRNA gets converted to protein, it will be called translation. This is all about the chapter. This is all about it. Now, in this, you will have to understand what is DNA. You will have to understand what DNA is made up of. You will have to understand what is mRNA, what is protein. That's all, students. Nothing more in this chapter. This is all about the chapter. Do not get panic. I will make sure that this chapter becomes very, very easy for all of you. Okay, very, very easy for all of you. So, we will start when we talk about molecular bases. Let's also understand what we are going to do in this chapter. In this part of the lecture. Students, first we will be understanding polynucleotide chains. What we will be understanding? Polynucleotide chains. That means what is our DNA made up of, right? What is our DNA made up of? Polynucleotide chain we will be learning. Now, other than that, we will also understand the discovery of DNA. We will understand the discovery. Next thing, that is search for genetic material. Search for genetic material. Right? How did they actually come, or how did they come across this DNA? How did they find DNA? What was the thing that led to the finding of DNA? All these we will be understanding in the class. Yes or no? Okay, we'll be understanding all this in the class. Then we will be understanding how are these nucleotides made? What are the components of the nucleotide? Right? We will be understanding what is the component of our nucleotide. Yes or no? What is it made up of? Okay. Then we will understand how is their bonding. Okay. How is their bonding? Do they bond with glycosidic linkage? Do they bond with phosphodiester linkage? All that linkages you will understand. Then we will understand how did they tell that DNA is the genetic material and not RNA. Why is RNA not the genetic material? So, we will understand the criteria for being the genetic material, or we will understand how DNA was told that yes, this is the genetic material with the help of some experiments. Okay, some experimental setups you will be understanding. This is what we will be doing today. Okay, I hope this is clear. Okay, so students, when we talk about DNA, I want all of you to understand few things. See, understand that DNA is the genetic material, but definitely it did not just come like that, right? It took some experiments to find out why DNA is the genetic material, why not RNA, why is RNA a single stranded, why is DNA double stranded. All this took a lot of time. So in this chapter, you should understand it like a story. Okay, think of it as a story. We started from the first step and now we have reached the final steps. Okay, this is what the chapter is. And this thing which I have written here is your central dogma. Very, very important. Questions can come from both also and for your NEET also. Central dogma. What is the central dogma? This is the deciding factor of your genetics. This is the center, like how we have the brain. No, this is the center of everything. Okay, shall we continue? Now, search for genetic material. How did they actually, bring about anything? How did they actually know that, okay, this is the genetic material, this is what we have to do with it? How did they come to this conclusion? Yes or no? Yes, okay. Students, you can see me clearly, no? Yeah. So how did they come to know about this is what we are going to find out. Okay, all of you share the link with your friends. I don't think all of you are sharing link with your friends. It's very bad. You should share link with your friends. Help them. Okay, so students, first genetic material, like in the sense DNA was not found. First, what happened was there was a scientist called as Friedrich Mischer. Okay, Friedrich Mischer. He was, trying to find out something, and he happened to find something called as nuclein. Okay, he was trying to do, experiment, and he found first ever thing, that is your genetic material, is nuclein. Okay, he found nuclein, and this discovery took place in around 1869. So he was trying to find something, he found nuclein. And then everyone was like, okay, this is something to do with our genetic material. See, there was no clarity. There was no clarity that this is the genetic material, but Friedrich Mischer understood that this had to do something with our, genetic material, something with our genetic material. Then definitely many other scientists also came behind them. You do not have a lot in depth about how did he find nuclein, all that is not there. Just understand Friedrich Mischer found nuclein, and they understood that this had something to do with the genetic material. Okay. Now, there was another scientist after that who is called as Altman. Okay, his name is called as Altman. Can you see red in the thing? Yes, you can see, right? Yeah, Altman. He found that this nuclein, which this Friedrich Mischer found, this nuclein had acidic property. What did it have? Acidic property. So he found that, okay, the nuclein that Friedrich Mischer found had some kind of acidic property. So then why don't I call this nuclein as nucleic acid? Okay, why don't I call this nuclein as nucleic acid? Okay, students, Friedrich Mischer found nuclein as a fluke. Like, how do you say fluke? Means, see, he was trying to do something with this pus cells and all that. And, WBC, phagocytosis, all this he was trying to work out, and suddenly he found something else, that is nuclein. Okay, so it was like a fluke that he found nuclein, but it definitely changed the entire, concept about, biology by finding this nuclein. And then Altman was like, this nuclein has some acidic property. So why don't we call it nucleic acid? This was the first discovery. Okay, so he found, in simple words, you can say he found the acidic nature. He found the acidic nature of your genetic material. Okay, acidic nature of nuclein, you can write, because it is still not the genetic material, nuclein. Okay, now when we talk about nucleic acid, they realize that there are two types of nucleic acid. One is your DNA and one is your RNA. Okay, so DNA is called as deoxyribonucleic acid. Yes or no? Deoxyribonucleic acid. Then RNA is called as ribonucleic acid. Yes or no? That's all. So DNA is called deoxyribonucleic acid. RNA is called ribonucleic acid. So from the name itself, you can see deoxy. That means one oxygen is less compared to RNA. Simple. DNA, deoxyribonucleic acid. What is absent? Deoxy. That means what? Oxygen is absent, right? Oxygen is absent. Okay. Yes, this is okay. So far good, students? Yes, okay. So this is all about it. Okay, this is how much they understood. And they knew that this DNA was double-stranded. They didn't know the double helical structure, but they knew this was double-stranded. And they knew RNA was single-stranded. Okay, students, if you want my notes, if you want all my handwritten notes, please follow me on Biology by Gopika, which is my Telegram channel. All the notes will be shared there. And if you want notes of your physics, chemistry, all of that, please do join our Vedantu NEET English. We will get all the subjects notes. Okay, yes. Fine. Now let's understand more about nucleic acid. Okay. So students, nucleic acid. Now full form. What am I talking about? The full form. I'm talking about DNA and RNA. Okay. So first we will talk about DNA. Okay. Let's do one thing. Let's write it on the top. Okay. Yes. Now let's write here DNA. Let's write here RNA. Yes or no? Okay, fine. Yes, I am writing the notes for you people, so please do copy down. Okay. Now students, DNA full form is what? Just now we discussed. Deoxyribonucleic acid. That's all, right? RNA means what? Ribonucleic acid. Okay. Now the structure. This is double-stranded. This is single-stranded. Yes, okay, clear so far? One is double-stranded, one is single-stranded. Okay, that was about it. Then students, location. Now what do you talk about location? Where is it present? Where is DNA and RNA present? DNA and RNA will be present in the nucleus of the cell. Yes or no? Yes or no? Okay, even though the synthesis is happening somewhere, the production or your this will happen will happen in the nucleus. Will happen in the nucleus. So RNA here, DNA is in the nucleus, no doubt in that. But your RNA is synthesized. Okay, it is synthesized. Okay, it is synthesized and stored. Synthesized and stored in the cytoplasm. Synthesized and stored in the cytoplasm. Important, it is synthesized and stored in the cytoplasm. Okay, students, what are they here? What are they telling about? Found in where and where? DNA is a genetic material in what? In almost all the organisms. Almost all the organisms. Okay, except for virus, right? Except for virus. Definitely lot of exceptions are there, but just as of now we will only write virus. And DNA is present where? In virus, your tobacco mosaic virus. Okay, there is a lot of examples given. Again, you can write this. Okay, this is clear. Now we will talk some things about DNA. Who is DNA? What is it doing? All this we have to know, right? Who is DNA? What is it doing? Okay. Yes. Okay. What is this DNA doing? All this we need to know. So DNA, we know it is a long polymer. Okay, it is a long polymer. Polymer of who? Polymer of who? That we have to know. Okay, 1 second. Yes. DNA is a long polymer of your nucleotides. Okay, what is it a long polymer of? Your, we can say, students, you can normally write polynucleotide, otherwise you can write deoxynucleotide. To be specific, you can write deoxynucleotide. So it's a long polymer of deoxynucleotide. We will understand what is nucleotide. Do not even worry. Just as of now, understand what is there in your NCERT. These all lines are from NCERT. Okay. I've kept NCERT with me, so I do not even miss one line. I will make sure that everything of NCERT is covered. Okay. So it is a long polymer of deoxynucleotide. Okay. Now students, length of the DNA. Okay. Next, very important thing, length of the DNA. Length of the DNA depends. It depends on the number of nucleotide. Yes or no? Depends on the number of nucleotide. Simple. Why, students? Because see, if your marks in your NEET depends on the amount you prepare. Yes or no? Simple. Okay, the amount you prepare will be directly proportional to the marks you get in your NEET. Same way, the length of the DNA will depend on the number of the nucleotides. Isn't it simple? Yes. So that's all. Okay, that's all. Do not get yourself confused. It depends on the number of nucleotide. Okay. Now, this DNA, I told you it is made up of nucleotides. Yes or no? From that time, I have been telling nucleotide, but I have not told what is nucleotide. So, nucleotide basically has 3 components. It has 3 components. These 3 components constitute together, bind with each other to form your polynucleotide or nucleotide as a component. Many nucleotide together forms your polynucleotide. Okay, so it has 3 components, and those 3 components are one by one we will write. Okay, first is this sugar. Okay, it can be ribose sugar, deoxyribose sugar. So we have sugar, we have phosphate group, and then you have your So here I have to write, 1 second. Let me write here. I will write here pentose sugar. So it is easy to remember, pentose sugar. Okay. And here we have nitrogenous base. Nitrogenous base. This is the 3 components of your nucleotide. These 3 people together combine to form your nucleotide. And then what did I tell you? I told you that many nucleotide, that means nucleotide plus nucleotide plus nucleotide, like that many nucleotide will form a chain, and that chain we call it as polynucleotide. Clear? So here in your textbook there is given as structure of polynucleotide, which we will be discussing. Okay, so far good? Okay, so far good? Yes. Yes. Okay. Now RNA, just a general thing. Okay, I'm not going in depth of RNA. We will just understand the general thing. Okay. So students, RNA, all of us know RNA. First thing we know about RNA is the full form, right? Ribonucleic acid. Then it was deoxyribonucleic acid. Here it is ribonucleic acid. Here, why it is called ribonucleic acid? Because one oxygen is extra here, right? So here we have the presence of oxygen. Okay, we have a presence of oxygen. Yes or no? Presence of oxygen. Then students, what do we know? It is single stranded. This also we know. It is single stranded. And there is also a theory or an understanding that the RNA was the first genetic material. Okay, it was the first genetic material that helped in the formation of your DNA, which is a more stable one. So even though RNA is not a genetic material for most of the organism, it is the first genetic material, initial genetic material, which actually helped in the formation of a more stable DNA. Okay, more stable DNA. Is this clear? So this RNA, when we talk about, RNA is a single stranded structure. It also has nitrogenous bases and all that. But now understand, this RNA, because it is single stranded, it does not hold the stability that DNA has. That is the reason why this is not a genetic material. Okay, why this is not a genetic material. So this so far good? Yes, and it is a genetic material in virus. So the question can come, RNA is a genetic material in what? Okay, the answer will be genetic material in virus. Okay, in virus. Yes. Okay. Good. Fine. So this is the general, students, just the general understanding. Now we will have to understand. I don't know if this is visible to all of you. Some editing happened, so I will just write it down again for you. I will give you the proper notes. Okay, I'll send you the proper notes on my Telegram. But as of now, we have to write with this notes because something happened, like it moved or something. Okay. Yes. Wait, let me see if it's visible. Right? If it should be visible. Yes, is it visible? It should be slightly visible, right? Slightly visible, no? 1 second, students. I think some editing has to be done. Yes. Okay, so look here, all of you. Yes, so there are some values given in your textbook, yes or no? So those values is what we are going to study. So students, lambda, okay, lambda 174, okay, lambda 174 has a number. So here what are they talking about? Bacteriophages, which is known as lambda 174, has a nucleotide value of 5386 nucleotides. I want all of you to focus here. It is mentioned nucleotides. It is not mentioned base pair. Okay, do you understand? That means what? They are single-stranded. So they are talking only about nucleotides. They are not talking about base pair. They are not talking about adenine and guanine binding together. They are not talking about that. Okay. So is this clear? Then students here, see here. Okay. Here next one you have is your lambda. Okay. Bacteriophage, which has around 48502 base pair. Okay, these are the values. This you have to by heart. There is no other way. Okay, this is 5174. I told lambda by mistake. 5174. This is lambda 48502. Okay. Next, what do we have? Next, we have 4.6. Who is 4.6? E. coli. E. coli is 4.6 into 10 to the power 6 base pairs. Okay. And last, whom do we have? We have haploid content, okay, of a human DNA that is equal to 3.3 into 10 to the power 9 base pairs. Students, now very important question is, it is mentioned haploid. Then what about diploid? Our body, what do we know? We know about meiosis, right? We know about meiosis. We know that it is reductional division. Reductional division. Yes or no? Reductional division. And during that reductional division, we have 46 chromosome. We have 23. Yes, we have 23 pair. Okay, this is how we say. Now students, here we have to see diploid. That means what? Basically multiply this 3.3 into 10 to the power 9 into 2. So you will get 6.6. Yes or no? You will get approximately 6.6 into 10 to the power 9. This is your answer. Okay, this is your answer. This is what you have to focus. Okay, these values you have to by heart at any cost. Okay. Now, when we talk about nucleotide, let's talk in depth about nucleotide. Okay, let's talk in depth about nucleotide. Is it visible? All the writings are visible, no? Yes, it is visible. Okay, students, now we will be talking in depth about nucleotides. What is nucleotide? What is it doing? All that we will be discussing. Okay, give me a minute by then. Yes, students, I will share the notes on my channel. Do not even worry. Okay, now just focus on the class. Just focus on the class. The rest assured. Okay, yes. 1 second. Not this. Okay, so students, nucleotide, I already give you a general idea. Okay, I already give you a general idea. Nucleotide is basically the constitution, like the unit that forms the DNA. So nucleotide has 3 components. This also did I discuss with you all? Yes or no? I discussed. Okay. Oh wait, where did it go? Now students, we will be discussing more in depth. Okay, we will be discussing more in depth. So students, when I talk about nucleotide, we have 3 parts. Let's do one thing. Let's write it this way. Okay, let's write it this way. Yes. Oh, flight takeoff went down. Okay, I think we can't write it like that. On the board I can write it, but here we will do one thing. We will just start writing this. Okay. Yes. So students, phosphate group. Right, phosphate group, first thing I told you. So we discussed phosphate group, pentose sugar and nitrogenous base. Now we are going to understand what is this phosphate group. Okay, this phosphate group, you have to know that this nucleotide has 3 components, out of which phosphate, sugar and nitrogenous base are the most important part. Okay, so let's also understand their diagrams as well, like the structure of it as well. So you have P, okay, OH, okay, OH, double bond O, and you have OH. Simple. 3 OH is coming, 1 double bond O is coming. This is your phosphate group. Okay. Now bonding, how it is happening, I will tell you. But as of now, just understand so much. Okay. Then the second component is your sugar. Second component is your sugar. In sugar also there is 2 types. Why? Because we have DNA and RNA. We are talking about the nucleotide that constitute everything, right? So we also have deoxy, and we also have ribo. Okay, ribose sugar. You can tell ribose sugar. Okay, easy to remember, ribose sugar and deoxyribose sugar. Ribose sugar. Now, when I draw this also, all of you focus here. Oh, yes. Okay, one second. It is chemistry, so we need little concentration to write chemistry because we are bio teachers. Yes, this is your structure. Okay, this is your structure. So here bondings will be there, right? So students understand here instead of a OH, okay, in a ribose sugar there will be a OH. Here we have just H. Okay, so the structure remains the same. Only thing, the O group will be there. Okay, I'll show you the difference. Here we have a OH. We have a OH. Okay, understood? This is the difference. So here you have a OH, here you have a H. So presence or absence of that O in your DNA makes it so special. Okay, next, last one, what did I tell you? Nitrogenous base. Yes or no? Last one I told you was nitrogenous base. Nitrogenous base. Students, chapter might look very difficult, but trust me on this chapter is very easy. Let's try to understand as easy as possible. Okay. Now students, nitrogenous base is basically two types. Okay, two people are there in nitrogenous base. First person is called purines. Second person is called pyrimidines. Purines and pyrimidines. Now there are other people under this purines, like purines is the head, there are again sub-peoples under this. Okay, so how are we going to remember this? Purine, first thing you have to understand about purine, it is a double ring structure. It is a double ring structure. Okay, this is a double ring structure. Very, very important. Now we have to draw the structure also. Let's draw the structure also. See, look here, all of you focus here. So this is a little, maybe it looks difficult in the starting, but it's not that difficult, students. First you draw a 6-ring structure. Okay, then from here you draw like this. Okay, don't do that. Yeah, just connect this. Okay, so now we have to start counting. So you have to always count from here. Okay, 1 2 3 4 5 6 7 8 9. Okay, 9. Always remember, when you draw your purines, you should always know that the purines will have 9 positions. Okay, 9 will be the number, so it will be a 9-ringed structure. Okay, it will be a 9-ringed structure. So whenever you draw purines, if you feel that the structure is somewhere wrong, you should make sure that you count it and you see that it is a 9-ring structure and it is a double ring. Can you see? 1 ring and 2 ring. Okay, and the number is 9. 9 positions are there. Okay, 9 carbon it is. I think 9 carbon we will write. So you will always remember, because 9 rings might be a little complicated for some people. So we will write 9 carbon structure. Yes or no? Now, more about purines is that, we will erase this pyrimidines. Okay, more about purines is that, students, to remember, how do I remember purines, right? How do I remember purines? You will remember pure as gold. How will you remember? Pure as gold. P stands for purine, A stands for adenine, G stands for guanine. So I told you, purine is the headmaster. Purine is the headmaster. Under the headmaster, many sub people will come. No, teachers will come. All this will come. So pure as gold. Purines. A stands for adenine. G stands for guanine. This is all about your purines. Okay. As of now, just understand this much. It's very important for you to remember adenine and guanine, because students study a lot of it for the exam and go and they forget. Is adenine coming in purines or pyrimidines? Is guanine coming in purines or pyrimidines? This confusion always keeps coming. Okay. So that's why you make sure that you remember pure as gold. Okay. Now we'll try to understand pyrimid. Okay. Now when I talk about pyrimidines, can you see me? Because I'm sitting here. Can you all see me? See the slide? Students, do not worry. I'll show you the slide. Okay. I'll write pyrimidines here. Okay. I'll write the pyrimidines here. Yes. So students, here pyrimidines. Pyrimidines, I'm writing it here so you people can see. Now pyrimidines, first thing you need to remember, it is a single, okay, it is a single ring structure. Okay, it is a single ring structure. There it was a double ring structure. So don't we have to draw this? Yes, we have to draw this. Very simple, like your purines itself, draw the first part, only the first part is the same. Okay, 6 carbon points are there. 6 carbon points are there. Okay, this is a single ringed structure. Now, when we talk about how do I remember pyrimidines, ma'am, because purines are also there, pyrimidines also there, how do I remember it, right? You will remember cut. You will remember cut. You'll be like, ma'am, there we have only 2, here we have 3. I'll tell you why that 3 has come. Okay, first C is cytosine. Next U is uracil. T is thymine. You understand? Cut. Cytosine, uracil, thymine. Now, out of that, your uracil comes in your RNA and not in your DNA. It is present in RNA, not in your DNA. Very, very important thing. So, thymine will be replaced with the help of uracil in your RNA. Okay, thymine will be replaced, okay, replaced with uracil in your RNA, okay, RNA. So in DNA, if thymine is there, in RNA what will be there? Uracil will be there. Okay, clear? Easy? Okay, very easy? Yes, fine. Okay, now students, when we talk about your nitrogenous bases, you have to know. So I hope that is clear. I hope that is clear. Easy, no? Let's do a quick revision. Nucleotides are made up of phosphate group, sugar and nitrogenous base. Phosphate group, you have P, 3 OH and double bond O. Okay, sugar, when you see, you have deoxy and ribose sugar. So in deoxy, 1 oxygen is absent. Look here, 1 oxygen is absent on the second carbon. Okay, and here in the ribose sugar, 1 oxygen is present. Okay, then nitrogenous bases are of 2 types, purines and pyrimidines. Purine is a double ring structure with 9 positions, and it is, how do you remember it? It's pure as gold. Purine, adenine, guanine. Okay, then we have pyrimidine, which is a single ring structure. 6 of them are there, and how will we remember it? Cut. Okay, cytosine, uracil, thymine. And thymine will be replaced by uracil in the RNA. That's all. Very simple. Now, students, nitrogenous base, you have to remember purines. Okay, purines. I told you first, let's draw the structure. Then only I'll be able to explain it to you. Students, I can easily use a PPT and teach you this, but I know that writing and teaching, it always helps. Okay, writing and teaching always helps, because students will be able to write the notes with the teacher, and they will also get the time to write and understand. Ma'am is also writing, even let me also write. Okay, otherwise I can just put the PPT and bring it here, but I'm not doing that. Okay, yes, this is the structure. Yes or no? Agreed, all of you? So let's start labeling. 1, 2, 3. Oh, one second. Yes, okay. 1, 2, 3, 4. Yes, here, where will it come? 5, 6, 7, 8, 9. These are the positions. Yes or no? Now, if you look here, out of these positions, students, 4 positions are nitrogen attached. Okay, what is it? N will be attached on 4 positions. Which are those 4 positions? 1, 3. Okay, so you should remember this way, odd numbers. Okay, all the odd numbers. Okay, so it will be 1, 3, 7 and 9. Okay, if you know chemistry, you know there is para, meta, positions and all. I'm not going to teach chemistry. Okay, 1, 3, 7, 9 will be nitrogen attached positions. Okay, nitrogen attached position. Okay, yes or no? Purines are what basically? They are all made up of your nitrogen based or your amino acids, right? So N will be there. N attached positions will be 1, 3, 7, 9. Okay. Now on pyrimidines, what will be the position? What will be the positions on pyrimidines? Let's see that also. So on pyrimidines, you have 6 positions, right? 1, 2. See, these are the positions. So again, you label from here always. 4, 5, 6. Out of the students, look here, your 1 and 3 will be nitrogen attached. Okay, N attached position will be 1 and 3. Okay, so you can remember it as odd numbers, you can remember it any other technique you want to remember, but just do remember that if any question comes any question comes about, which is the structure, which is the position where nitrogens are attached on these purines and pyrimids, you should always know it is 1379. Okay, 1379. Okay, clear? Yes, clear. Fine. Okay. Now, students, look at the structure. This is how the bonding will happen. Now, if you look here, we have a phosphate group, we have a nitrogenous base, and we have a pentose sugar. Out of this, always remember that the phosphate group plus the sugar forms the backbone. Forms the backbone of the DNA. Now, what do I mean by backbone of the DNA? Do you all understand backbone of the DNA? Yes, backbone of the DNA means this is the one who will be there to form this entire double helical structure. Entire double helical structure. The backbone is your phosphate group and pentose sugar. Then you will be like, ma'am, what about nitrogenous base? Students, nitrogenous base always protrudes. Okay, protrudes like this, like this. Like this they will be protruding. Like this they will protrude inside to make sure that they are in stable positions and they are bound to each other to form a double helical structure. Okay, to form a double helical structure. Clear? This is all about it. Okay, so you should understand phosphate group, pentose sugar, they bind with each other. Okay, they bind with each other. Okay, yes, fine. Yeah. Now this is the basic nucleotide. We understood the structure. I have put the structure here in case you people want to understand. So here you can see, students, how the binding is happening at the 1, 3rd, 7th and 9th position. There is nitrogen. Okay, this is at extra attachment. You do not count that. Okay, do not count that. This can keep changing depending on the type of nitrogenous bases. If it's adenine, okay, if it is guanine, okay, if it is cytosine, and if it is thymine. Depending on that, this one will keep changing. So you should always see that there is always N. So this is a purine, clear. So it is an adenine or it is a guanine. This one will keep changing. Okay. So you can identify a purine, but you do not have to identify which type of purine it is. Is it adenine or thymine? That is not necessary. But understand this is a purine. How will you understand this is a purine? By looking at there are 4 places where nitrogen is placed. Okay. Clear? 4 places where nitrogen is placed. Good job. Good job. Okay. Fine. This much clear? Yes. Now, students, look here. Again, purine. I have just put in case you people have a doubt. So here we saw, see last structure. Here we saw NH2. That means this was a guanine. This was a guanine. Now here if you see, okay, here if you see purine, this is how purine structure is. Again, I told you the positions. See 7, 9. So here, from here they have labeled. Okay, so I have drawn it reverse, right? I have drawn it reverse. Now here if you see, adenine will, NH2 will be on the top, here guanine, here NH2 will be here. But it's not necessary for you to know, but just look at it. Just look at it. In case a question comes, you should always be prepared. You should always be prepared. Yes? Here pyrimidines also have put the structure for all of you to understand. Okay, pyrimidines also have put the structure. You can see cytosine is there, thymine is there, uracil is there, right? Depending on that, the structure also is changing. See here you can see H is there here CH3 is there, here H is there. Now you have to spot the difference between these two. Okay, if you see here NH2 is here a double C is there. So the structure varies. Okay, structure varies. Now, this is a diagram given in your textbook, right? Yes or no, this is a diagram given in your textbook. So, what does this diagram indicate, students? Understand, the fifth, we call it 5-N to 3-N, no? How do we call it 5-N to 3-N? On the fifth, that is on the 5-end, that means that one end, the phosphate will be free. That is the 5-end, phosphate will be free. And on the 3-N, always an hydroxyl will be free. Okay, so the free ends indicate the 5- and the 3-. So if on one end phosphate is free, I will call that at the 5-end. If on the other end hydroxyl is free, I will call the 3-end. Okay, now why is this dash mentioned? Okay, why is this dash mentioned? Is because, see students, now example, this is a very it's not given in any of your textbooks, this is just an additional information. Now imagine this is your pyramid. Yes or no? This is your pyramid. Pyramid also has 1, 2, 3, 4, 5, 6 numbers. Yes. Here, if I don't mention 5-, I will not understand that it is the end of the DNA. If I just put 5, that means it could be 5th of the pyrimidine or it could be the 5th of the adenine or purine or guanine, whatever. right? So to make that difference, during the labeling, to make that understanding easier, they have put phi dash. That's all. There is no big significance for it. Just understand these numbers and these numbers should not collide with each other. That means nitrogenous base numbers and the chain numbers should not get confused. That's the reason they have put that dash. Okay, phi dash into 3 dash, and here you can see 3 dash into phi dash in. So if you look here, students, like I told you, your phosphate and sugar is making the backbone. Yes or no? It's making the backbone. Now, if you see, if these people are making the backbone, you can see these people are protruding in the between, right? They are holding it together. So, if it is a double helical structure, the middle portion is occupied by your nitrogenous bases. Okay, nitrogenous bases. Yes, clear? Yes. Now, we are going to understand the bonds. Okay, very little topic it is, but we will understand the bonds. Okay, yes. So students, when we talk about the bonds, we have to understand little complicated, but we will try to make it as easy as possible. I told you, first let's draw the phosphate. Like how I just showed you. Okay, now this is a phosphate group, yes or no? Now let's understand. I told you phosphate and sugar together forms the backbone. So first we are going to try to understand the backbone. We'll draw it with different colors so there is no confusion. Okay, now we will draw sugar. This is our sugar is. Yes or no? Right, this is a sugar. Okay, now in the sugar also, let's put all the parts. OH, H. Now imagine this is a DNA. If it is a DNA only, then H will come. Okay, please be careful about that. Okay, here also we put a H, and here we will put CH2OH. CH2OH. Now students, look what is going to happen. First thing will be dehydration will happen. What will happen? Dehydration. That means this H molecule with this OH will get removed. One water molecule removed. Okay, now we will also label what we have written. This is a phosphate, this is a sugar. Yes ma'am, these two are the sugar and phosphate, and these two people will form the backbone. So how do they form the backbone? They form the backbone first by losing one water molecule. Yes or no? Is this clear? Now, phosphate is there, sugar is there. So, I will want to call this bond. I have to label this bond. Yes or no? I want to call this bond as phosphoester bond. Can I call it phosphoester bond? Phosphoester bond. Students, phosphate is there. Okay, phosphate is there, sugar is there. Always remember, phosphoester bond is formed between your phosphate and sugar by the removal of one water molecule. Okay, simple. Okay, simple. So I told you that there is always a confusion, right? That's why they have put for this sugar also, the labeling is always going 1 dash, 2 dash, 3 dash. Okay, okay, do not worry about that. Now, I have to bind this sugar with a nitrogenous base. Okay, I have to bind the sugar with a nitrogenous base. Now, how do I bind the sugar with a nitrogenous base? Let's take a different color. Okay, now I will draw a purine. Okay, I will draw a purine. Very easy. Once you know the structure, no, the bonding is very easy. I am just explaining you with bonds. Usually everyone just tells you, like, you know, just tells that this is a bond, but oh, did I put ulta? 5, 6, yeah, inside. Students, this 5 should come inside, otherwise you might get confused. Okay, so we will put it inside. I don't want any confusions to my students. Okay, 5, 6, 7, 8, 9. Now I have to bind my sugar with a nitrogenous base, then only completion will happen. No, I'm trying to form a double helical structure, right? So how do I bind it, students? Very simple. This, from here, there is a bond going. Wait. Okay. Yes, there is a bond going, and let's draw the bond with another color. There is a bond happening. There's a bond happening between this. Okay, that means the bond is happening. Okay, students, I told you this 1 dash, 2 dash, 3 dash and all. No, that is also for your sugars. so that there is a difference in there is no confusion. See, if I put here also one, then how will I understand which part of the sugar is binding with which part of the pyrimidine or the purine? So that is why we have made the labeling 1 dash, 2 dash, 3 dash, 4 dash, 5 dash. So understand first one of the sugar and the ninth one of the purine will form a bond. Simple. The first carbon of the sugar and the ninth position of your purines will form a bonding. That bonding I want to call it. Okay, that bonding I will call it as your. Okay, here remember sugar is there. So it is called as 1,9 glycosidic linkage. Okay, this is the linkage. Understood? So how will you remember it? Understand there's a sugar and a purine, so you think of it like sugar is glucose, so glycosidic linkage. And where is the position? 1,9. Okay, 1,9 is the position. Okay, this is how the bonding will happen. So let's do a quick revision. A phosphate is there, a sugar is there, a purine is there. Now I've taken a purine, it could be a pyrimidine also. A purine is there. Okay, now I've taken a purine. Now imagine they have to bind with each other, right? Before that, students, let me also tell you this very, very important thing is sugar with your purines or sugar with your, nitrogenous base. I'll put nitrogenous base in the bracket. Nitrogenous base forms your nucleoside. All of you would have heard about nucleoside. That means you're pushing one person to the side. Whom are you pushing to one side? You're pushing phosphate to one side. Okay, so nucleoside means pushing one person to a side. Okay, nucleoside. Okay, so that is your, always remember nucleoside, pushing one person to the side. Okay, this is how you're going to remember. So students, let's revise the bonding very quickly. So we have phosphate, we have sugar. So let me know in the comments if I made this chapter easy for you. Okay, if you have watched this lecture, do not forget to comment, ma'am, you made the lecture easy, difficult, whatever you want to comment. I mean, good things, good criticism is accepted. Okay, yes. So students, look here. First we have a phosphate, then we have a sugar, then we have a purine or a nitrogenous base. This is basically our nucleoside. Yes or no? This is our what am I writing? Nucleotide. Yes or no? So students, nucleotide, we have these three people, they have to bond with each other, they have to bind with each other for forming the double helical structure. First we had a phosphate group. Phosphate group has to bind with a sugar. Now phosphate group will bind with the sugar. How? It will bind, this OH of the phosphate will bind with here OH and remove one water molecule will be removed, forming a phosphoester bond. Okay, a phosphoester bond. Then we have sugar with a purine or a pyrimidine. So sugar will bind with the first carbon of the sugar. It will make a bond by binding with the first carbon of, first carbon of the sugar to the ninth carbon of the purine and form a glycosidic linkage. So two bonds we discussed here. First is your phosphoester bond. That is between who and who? Phosphate. Obviously, phospho means phosphate should come with sugar. Then second we have glycosidic linkage. Between who and who? Between sugar and your nitrogenous base. Be it any nitrogenous base. This is all about the bonds. Here you can remember 1,9. Okay, this will change depending on which type of purine and pyrimidine is binding. Here students, dehydration. One water molecule gets escaped for this bond to happen. That's all. Bonding became very easy, no? So this is all about your bonding. Okay, simple, dimple, easy. Yes. Okay, easy. Now students, one second. Yes. Okay. This much, I hope all of you are writing your notes. I know it's a little small, but it's visible. Okay, it's visible, I hope. Yes. Yes, it is visible. Fine. One second, students. Yes, okay. Yes, this much clear. Bonding is done. Okay. Here I have mentioned the same thing. Okay, look here, students. Here you can see the bonding as well. See, this 2 is binding. Here one water molecule gets removed. This is a little more complicated, but otherwise if you see as a normal, you will understand phosphate and sugar. Here we will get a phosphoester bond. Phosphoester bond, and here you will get a glycosidic bond. Okay, glycosidic bond, glycosidic linkage. Now all of you understood, no, nucleoside, you're pushing the phosphate away. You're pushing the phosphate away. Okay, clear? Done. We are done. Okay. Now we are going to get into the discovery of your double helical structure. Okay, very important, discovery of your double helical structure. Okay, so students, this discovery is not any small discovery. It's a big discovery, right? It's a big discovery, because still that they didn't know if DNA was double helical, is DNA stranded. They didn't know anything about it, right? They didn't know anything about it. So it was definitely a big thing in the history itself. So when did all this happen? What happened? All that we have to understand. Yes or no? For that to understand, you should know 2 people, Watson and Crick. And if you understand that, you will understand the whole thing, and you will also have to remember Chargaff. So double helix structure. Okay, double helical structure. This double helical structure, the discovery was in the year 1950 to 1963. Okay, this was 3 years they took, 1953. Okay, around 3 years they were trying to find what is the structure of DNA, or how did the structure look like. But students, how did they find it? They found it with the help of X-ray diffraction. How will you know how your bone broken? How will you know if your bones, your metacarpal phalanges are there? What happens if the phalanges are misplaced, or anything movement has happened, or your patella is moved this side. How will you know? You will know by an X-ray. You will understand that my bone has got dislocated, ligaments have torn, or anything. You will understand. Same way, with the help of X-ray diffraction, they understood. With the help of X-ray diffraction, they understood that the structure, that means the DNA, is a twisted structure. Okay, a DNA is a twisted structure. So this helped them found the DNA structure. So students, to be frank, Rosalind Franklin was the first person to find it, but very sad that she did not get recognized for her work. She did not get recognized. Actually, Franklin, that is your Rosalind Franklin, was the first person to do an X-ray diffraction to find out the double helical structure, but somehow Watson, Crick and Wilkins got the recognition, but Rosalind Franklin did not get the recognition. It could be also because those times society was not very supportive of women the way now it is. Okay, now also there are still people who are not supportive of women and who talk very bad about women, but then still, if you see, generally 90% of people are very supportive of women. They give the kind of jobs and they give the kind of recognition, but those times these things were not very very accepted. It could be one of the reasons why she did not get recognition. Okay, and it's a sad thing that she did not get recognition, because she really helped in this process. So who and all was there? Watson, okay, Watson, Crick, Franklin, 4 people. Okay, I'm writing Franklin also. Franklin, Wilkins, okay, Watson and Crick, Franklin, Wilkins, all these people were the reason for finding the double helical structure. Double helical structure. Okay, now this double helical structure, yes, they did find it, but then after the double helical structure was found, they understood one more thing, that this nitrogenous bases are the reason why this double helical structure is actually present. Nitrogenous bases means who and who? Purines and pyrimidines. Purines and pyrimidines. They understood that these people are the soul of our DNA. They are keeping the relationships of the DNA intact by holding each other together, by binding with each other, right? So adenine, we know two people are there, A and G, and pyrimidines, we know there is C and T. Now, how do you remember the binding? Who is binding with whom? What is happening with each other? Students, you can remember this way at Gopika ma'am's class. So A will always bind with T with 2 bonds, 2 hydrogen bonds, and G will always bind with C with 3 hydrogen bonds. Simple. Okay. So at Gopika ma'am's class. So ma'am, you keep it in your mind only, okay? Ma'am, you keep it in mind. But at Gopika ma'am's class, you can remember to know the binding. Because again, I told you, when you're studying so much for the exam and going, there are chances that you get confused. Even sometimes when I'm teaching some things, I get confused, right? It is human tendency. Our brain is made in such a way that it does not have that long memory to hold the small things. Yes, if something funny, sad, happy has happened, yes, we can accept the emotions and we will remember it for a longer time. If someone has insulted us, if someone has complimented us, we remember for a longer time. But small things, if you do not go back and revise, our brain will not remember it. So there are chances that you can get confused in the exam. So always remember at Gopika ma'am's class. This is how the binding happened. So yes, they understood that these people are keeping this together. Then there was one more person called as Chargaff. Chargaff was like, wait, I know that they are binding together, but what is the ratio of their binding? What if like one person is more, one person is less? What is actually happening? So Chargaff told a rule that the number of or the ratio of purines, okay, ratio of purines will always be equal to the ratio of pyrimidines. Ratio of pyrimidines. See, in a class, if there are say 10 students and I'm asking all of you to make a group of 2, right? If I'm asking you to make a group of 2, 5 groups will be formed. But imagine at the same time I'm telling make a group of 3. One person will be left out. So that cannot happen, right? How many adenines are there? That many thymines should be there. How many cytosins are there? That many guanines should be there. Otherwise, how will the binding happen? So he came up with a rule telling that the ratio of purines will always be equal to the ratio of pyrimidines. Now here is a catch. Here is a PYQ question that can be asked, and most of the people never ever tells this and they forget about this. Simple, students, I will tell you what it is. A plus, so we have adenine and thymine. Yes, do agree to it. Purines and pyrimidines will always be equal. Okay, so that means the number of purines, this is purines, to the number of pyrimidines will always be equal to 1. Yes, agreed, ma'am. Yes, we do agree it. If I have to write it in another way, I can write purine upon pyrimid by pyrimidine will be always equal to 1. Yes, ma'am, we agree to this. This is what Chargaff told. Yes, totally agree. But the catch is here. The PYQ can be here. A plus T by C plus G. Do this has to be equal to 1? No, it does not have to be equal to 1. It does not have to be equal to 1. That means, what am I trying to say? I am trying to say that the places where adenine and guanine is there, like inside, like wherever adenine and thymine is there are places which are adenine thymine rich and there are places which are guanine cytosine rich. Okay, when you learn later, you will understand mini satellite area, micro satellite area. That means the number of GC in a DNA will always be more. Number of GC in a DNA will always be more than the number of 80. Okay, number of 80 will be less. Okay, they say that around 60% is GC in our DNA and only 40% is 80. Is this clear? That means adenine thymine sites need not be equal to guanine cytosine sites because they both are not binding. There will be areas where adenine and thymine is high and there will be places where guanine and cytosine is low. So those kind of mismatch can happen. So here is the PYQ question that can come asking that do purine, that means do adenine and thymine have to be equal to guanine and cytosine? No, the answer is no, because there can be regions where adenine and thymine is more and guanine and cytosine is less, and there are regions where guanine and cytosine is more, adenine and thymine is less. Okay, but always remember purine and pyrimidine has to be equal, otherwise binding will not happen. But the amount of how much these two will be there, that does not matter at all. Very, very informative, very important PYQ type of a question. Clear? Okay, clear? Yes, clear. Yes, got it, fine. Okay, understood? Yes, good job. Now students, next topic, salient features of DNA. Okay, salient features of DNA. Now what is the speciality of DNA? What could be the, what is the very, good thing about DNA? All this we have to understand. Okay, all this we have to understand. So that is what we are going to understand. I hope Chargaff rule is clear, then double helical structure is clear. Okay, now we will understand some very important features or salient features of your DNA. Yes, ready all of you? Okay, after that we'll take a quick break and then we will continue. Okay, then we will continue. Yes, did I teach everything? Yes, I did teach everything. Okay, fine. Students, here only little theory part. After this, it is like a story. After this, you can know it is just like a story. Okay? Yes. So, what is the salient features of DNA? That is very important. Okay? First salient feature of DNA is that sugar and phosphate forms the backbone of DNA. Sugar and phosphate forms the backbone of DNA. This we already know. Okay, and the strands of the DNA. Okay, the strands of the DNA. Students, when we told double helical structure also, I forgot to tell. Double helical structure, you always know that one strand, if it's going from 5' to 3', another strand will go from 3' to 5'. Right, they are complementary to each other. They will not be the same, then same, different, no? So they won't be, they will be 5' to 3', 3' to 5'. Okay, yes or no? So they are polarity, the polarity of the strands. Polarity of the strands will be how? 5' to 3' and 3' to 5'. Okay, anti-parallel. They will be anti-parallel. They won't be parallel. If they are parallel, then what will happen? Story khatam. They will fuse with each other and then gone. Right? They will not fuse with each other also. Yes, clear? Yes. Okay. Fine. Sugar phosphate is clear. Polarity is clear. 5' to 3'. So they are always, one more point I'll write here. They are always anti-parallel. They have anti-parallel polarity. Otherwise, how will the binding happen? No? Anti-parallel polarity. Clear? Next thing. The two chains are wound around. Right? They're wound around in a right-handed fashion. Right-handed fashion. There is left-handed fashion. You will know different types of DNA is there, A, B, Z. In that there will be left-hand and right-handed fashion. As of now, this DNA which we are studying is right-handed fashion DNA. Okay. And students, what do we know about DNA more than this? The pitch of the DNA. Pitch of the DNA is always 3.4 nanometer. 3.4 nanometer. Okay. Then we have to know the distance between the base pairs. Distance between the base pair. That means, see, one base pair to the next base pair. What is the distance? Okay, it is again approximately 0.34 nanometer. Okay, 0.34 nanometer. And how many base pairs will be there in one turn of DNA? Okay, one turn of DNA will have around 10 base pairs. Clear? So one turn like this, right? One turn of DNA. This is the first turn. Can you see my hand? This is the first turn. This first turn there will be how many? 10 base pairs will be there. Okay. So then every turn will have 10 base pairs. Clear? So these are the salient features of DNA. Then you can also write the strands are complementary to each other. You can write how many other points you remember. Okay. They are complementary to each other. Then you can write about Chargaff rule. Complementary to each other. You can also write about the Chargaff rule. You will get marked for this. So do not hesitate. Whatever you know about the DNA's, very special character, please do write it in the exam. Do not hesitate to write. You will get full marks. Okay, you will still get full marks. These are the important numbers. Okay, students usually get confused. So students, distance will be 0.34 nanometer, pitch will be 3.4 nanometer. Okay, done. Write up 2 times. Okay, yeah, okay, okay, 1 second, yes. So students, we will take a 2 minutes break. Okay, I mean not 2 minutes, we'll take a 7 minutes break. Okay, my throat is paining, so I'll just have something hot and come. So we will be back at, okay, all of you be back, take a quick break and come. I know all of you will be hungry also, take a break and come. Lunch break I'm not giving, but yeah. Just a small break. After that, we will do central dogma and we will come back. We will finish and we'll do the second part also. Okay, 2 p.m. all of you be back. Okay. Yes. Okay. Take a quick break and come back. Fine? By then, share the link with your friends. Ask them to also join. Okay, ask them to also join. Because it will be very helpful if you are doing a board revision. students, are you all back? I'm back. Hello students, are you all back? Yes. Okay, let's continue. Okay, student, now we are going to do a very, very, very interesting topic, okay? And if my pen vanish. 1 second, students. Vanished. Okay, let's start the class. Okay, central dogma I already introduced in the starting of the class, right? So you already know about it. It's not very difficult to understand now. So still we will do 1 second. So central dogma means these are the deciding factors. Like this is the, the ruling factor of the genetic material. The ruling factor of everything is your central dogma. So central dogma means what is entirely happening in genetics is central dogma, right? Basically, what did I say? I already told you this. So DNA to mRNA, mRNA to protein. This is central dogma. Now, each step has its own name, each step has its own importance. Yes or no? Now, when DNA forms its own copy from the parent DNA, from the template strand, it is called as replication. And DNA replicates in a semi-conservative pattern. This also all of you know. Now, DNA to mRNA is called as what? Transcription. It's called as transcription. mRNA to protein is called as translation. Again, students, understand these are not just like, there are a lot of steps into these, right? Lot of steps into these. Now, here what is happening is DNA will form its own template. Okay, DNA will form its own template with the help of your semi-conservative method. Now, imagine this is the template DNA. I can only show it with the help of 2 different colors now. So, this is how your DNA will replicate. Semi-conservative method. That means it will be hybrid, right? It will form its own hybrid. This we will study in detail, but just understand this much now. And then DNA will form mRNA. That means DNA is a double-stranded structure. That double-stranded structure will form a single-stranded mRNA, right? Here also RNA processing is there. Capping is there, tailing is there. Again, a lot more complicated process, but as of now, understand that a double-stranded DNA formed a single-stranded mRNA. Okay, this single-stranded mRNA will form protein chain. That is your polypeptide, not polynucleotide, polypeptide chains. Yes or no? What will it form? Polypeptide chain. And it is not happening just like that. It is happening with the help of your genetic code, genetic codons, your tRNA, ribosome. So many people are taking part in this. Okay. Now, at the same time, reverse can happen. What do I mean by reverse? RNA can get converted. RNA can get converted into DNA. Yes or no? Which is called as reverse. Okay, which is called as reverse. 1 second. This is called as reverse transcription. Reverse transcription. Okay, this is possible by whom? This is possible by your HIV virus. Okay, AIDS causing virus, right? Human immunodeficiency virus, which causes AIDS. That is the reason why AIDS is not so curable, because why? How much ever you try, you will not be able to control the multiplication. So students, HIV virus, what they do is, this HIV virus will go and bind with your CD4 receptors. And these CD4 receptors are present on your macrophages, T cells and dendritic cells. So this HIV will go and bind with the help of your G120, that is the protein which is present on this HIV structure. It will go bind there, it will go inside, it will manipulate our nucleus to theirs. That means it will tell our genetic material to start forming their own proteins. That is the reason why we cannot control the replication of HIV virus. Okay, so HIV virus have a special capability of converting their RNA into DNA and confusing our macrophages to form more of HIV. Okay, so our nucleus will get confused because it will put its genetic material and it will confuse us and it will start forming viral proteins instead of forming our proteins. So this process where mRNA gets formed into protein, here what is going to happen, it will start forming viral proteins, and this is the reason why HIV is not in our control. Okay, or AIDS, right? AIDS causing germ is not in, virus is not in our control. Okay, if this much is clear, can we proceed? Okay, so this is basically your central dogma where you have transcription, translation and replication. Okay, now students, we all understand about the fact that our DNA is very long. Okay, approximately it is 2.2 nanometer long. Now such a long DNA, how am I supposed to put into a nucleus which is super small? Yes or no? Understand? Now imagine your DNA is a long thread. Now how am I supposed to constrain? I have to roll this thread and put it into a very small nucleus, or I have to put it into a very small dabba, right? Like a small, box. Now for that to do, I need packaging. I need packaging materials, right? So I am giving all of you a very long thread and I am telling you, do one thing. I have to put this thread into this small box, and this thread is around 2.2 meters. Sorry students, I put nanometers, 2.2 meters, okay? 2.2 meter long. Such a big thread. I am telling you, put it into a small box. You'll all think of an idea. Maybe the idea is, some people will be like, well, let's do one thing, we'll tie knots. Or some people will do one thing, we will just we'll put a small, like a ball kind of a structure, and we will roll it around it, or we will roll it around it, right? So like that, when we were thinking, they understood that there are specialized proteins called as histone proteins. Okay, specialized proteins called as histone proteins, which are positively charged. Very important thing to remember. It is positively charged and it will help in binding the DNA around it. The DNA is negatively charged, so these both will bind happily together. Now, how does the binding happen, right? How does the binding happen? So, histones are specialized protein that helps in the packaging of DNA. The process is called packaging of DNA. Okay, so histone protein, students, they are of 5 types. We can tell 4 types. Okay, 4 types are there. 4 types. 4 types of proteins are there. Okay, 4 types are nothing but, I tell you why I told 5. See, 5 are there, but then types we cannot tell that much also. So students, we will start with H1 protein, H2A, H2B, H3 and H4. If you look, there are only 4 types because these are just variants, H2A and H2B. But if you tell, there are 5 main histone proteins helping for this binding. Okay, out of that, this H1 is actually not helping in the packaging part. It is only helping in holding all this together. Now I told you, now this is a DNA. Yeah, I think this is not blurred, you know. 1 second. So, students, this is your DNA. Okay, this is your DNA. Now, I am telling you, this is your histone protein. Now, I am telling that you bind around it. You wound around it, right? Like this. You wind around it like this. Keep on you going, and you wind, and make this 2 meter long DNA into one bead on a string. Okay, like this, many will be there. 1 histone protein will be there, 1 more will be there. Like that, they will keep forming, and they will keep this 2 meter DNA into the nucleus. Clear? This much is clear. Now, the question arises, how are they formed? Like, how are they in the packaging pattern? How are they in the packaging pattern? Is it one histone with one strand? How is it, right? So, this H1 protein, no students, they act like linker. They act like linker protein. What is linker protein meaning? I will show you. As of now, understand. Now, all these histones will form something called as histone octoma. What will they form? Histone octoma. Octoma means 8. Yes. Now you will be like, ma'am, only like 5 proteins are there, how will they form 8 structures, right? So I will show you. Now we will use different colors to identify, like to understand it, different colors. Yes, okay. Yes. So we will put here, this will be H2A. Wait, we will draw bigger structures. Okay. Yes. H2A. This is 3D. Okay. This is also H2A. Now next protein we will draw. H2B. Do you understand? Each of the protein is making two of them. That is how they are forming the octamer. Okay. Now let's draw H3. Okay, and we will form what? Last one is H4. Got it? So they will basically form an octamer. That means what, students? Each protein will take up 2. Each protein will be doubled. 2 of them will be forming together. Now if I want to wind my DNA around it. Okay, I want to wind my DNA around it. So this is the DNA thing. Okay, it is going around and we cannot see it. It is not 3D. I cannot draw 3D. But it is going like this and it is coming like this. Okay. This is how my DNA will look. Now to hold all of these people together, right? We have to hold all of them together. We will use the linker protein. This is your linker protein. So H1 protein will keep all of these people in place. Is this clear? Okay. So this becomes your H1 protein. Okay. Each of them will have 2, 2, and 2. This is how they form an octamer. This is how they form an octamer. So 8 of them will be formed. So students, understood? H2A, H2B, H3, H4, all of them will be doubled to form like a cube-like structure, right? Like an octamer. So this entire structure with the help, that means what? I can call with the help of DNA and histones all together. Okay, all together. One second. DNA plus histones together, I will call the structure as nucleosome. Nucleosome. Okay, understood? DNA plus histones will be called as nucleosome. Now students, not one nucleosome will be there. Many nucleosomes will be there. So how will they look like? How will they look like? They will look something like this. So one nucleosome, DNA, nucleosome, DNA, nucleosome, DNA. Like this, it will look like, it will look under the microscope like beads on a string. Beads on a string structure. Clear? So this is how the packaging of DNA happens. So we have specialized proteins which are positively charged. These proteins will wind around or help in the winding of the DNA around them, and there are around 4 types of protein, 5 types also you can write, no problem. H1, H2A, HB, H3 and H4. These ones, out of which the H1 will be linker protein, which will bind everything together, which will keep everything together in place. And each of them will have 2 of them to form a histone octamer, and DNA with histones is called nucleosome, and many nucleosome will be there on a 2.2 meter DNA, which will look under the microscope like beads of string. Okay, one image is given in your NCERT for all of you. See, they will look like beads on a string structure. This is your packaging of DNA. So that is how they make this 2.2 meter long DNA fit into the nucleus. Okay, now you have to know more about histones. Okay, you have to know more about histones. Histones are proteins. Yes, I told histones are proteins. These proteins, this histones is made up of mainly 2 proteins called as lysine and arginine. Lysine and arginine. Okay, lysine and arginine. These are the proteins. Histone is made up of positively charged protein. These proteins are made up of lysine and arginine. These 2 are what, students? Basically amino acids. Proteins are made up of amino acids. So these are the main amino acids which are present in histone proteins. Clear or not? Okay, clear or not? This is how such a long 2 meter DNA will get fit into a small nucleus. Okay, a small nucleus. Okay, clear. Fine? Okay. Now, very important. One nucleosome has 200 base pairs. This is a question, okay? One nucleosome has 200 base pairs. Then how many will be there in humans, right? How many will be there in humans? We know that haploid content of your DNA, okay? Haploid content in your human DNA is around human DNA. Students, keep all this mentioned. We can expect all this as PYQs because most of the students reading NCRT will not be able to figure this out. So that is why you need a teacher. That's why you need a teacher who will make you understand, right? So these things can come as PYQs. So please make a note of it. Students, we told that haploid DNA in the textbook is given 3.3 into 10 to the power 9 base pairs. But we know that humans have diploid, right? Humans are diploid. So diploid also we did the calculation. So diploid of human DNA, right? Diploid content on the human DNA will be, I'm not writing content, but I hope you understood. It will be 6.6 into 10 to the power 9. So they are asking that one nucleosome, okay, one nucleosome, that means with your histone, octamer and DNA, has around 200 base pairs involved. Then how much will 6.6 have? That is what the question is. How much it will be, right? How much it will be? So we will call it x, no? So this you will cross multiply, right? So how much it will be? Not cross multiply, division will happen. So it will be x is equal to, say 1x. You don't have to write 1x, just write x. This we label it as x, okay? X is equal to what, student? This will go 6.6 into 10 to the power 9 divided by 200. Right? Right? So when you calculate, you will get somewhere around, I think 3.3 into 10 to the power, okay, 7 base pairs. So this is how many base pairs are involved. Okay, this is how many base pairs are involved in your diploid human content. Okay, human content. So students, you can just do the multiplication. You can do this 2 into 10 to the power 2 and then take it on the top, minus it. Just so if you want me to do the maths, I will do the maths. 10 to the power 9, right? Here what I'm going to do, 2 into 10 to the power 2. So when this goes up, what will happen? Minus will happen, no? So here it will become 6.6 into 10 to the power 9 into 10 to the power minus 2 divided by 2. This and this I will cut. How much it will be? 3 2s are 6, 3 2s are 6. Okay, and here it will be how much? 3.3 into 10 to the power 7. Okay, plus 9 and minus 2, 7. This is how the maths is. Okay, so this is how much nucleosome will constitute for your diploid DNA. Okay, then students, in your textbook one more thing is mentioned, that is your non-NHC is mentioned. Okay, what is NHC? That means non-histone chromosomal protein. What is it? Non-histone chromosomal protein. Non-histone chromosomal protein. Now this non-histone chromosomal protein is what, students? Basically understand, this DNA will keep winding. That's how in the prophase, when you're looking under the microscope, if you have done this onion root tip, or some people also have done, I think, skin. Okay, skin and all that. If you have done mitosis, meiosis, if you have tried to see mitosis under the microscope, you would have seen a prophase, right? All of you would have seen a prophase where there will be a circle inside like this, pink color you will stain, and then you will see like this one stage, right? Where everything will be like a mesh, right? Now, how does this happen? It is basically your DNA will keep moving. That means winding, and they will finally form your chromatin to your chromosomes. Yes or no? Now students, you should understand one thing, that this non-histone chromosomal proteins are actually going to help when there is higher requirement. That means higher condensation should happen, no? For higher condensation, that time we will not, like, histone proteins will not be enough. That time we will use non-histone chromosomal protein. That means in higher organism, when there is maximum condensation, that time we will use, just like, like now, so much drama one student is making in the class, like fighting, shouting and all that. He is not listening to the class teacher. Then what class teacher will do? Class teacher will call principal, or class teacher will call HOD, right? Like that only. More energy we need, more influence we need. That time we will call this people. Okay, that time we will call this people. Okay. Is this clear? Yes? Done. Okay. Done? Yes. Fine. So that is your high condensation. Okay. Now students understand this one also. Now look here. Or if you look here to this diagram, you can see 2 meter DNA is there. Nucleosomes. Can you see how it's winding? See, one bead is there around the DNA. This keeps going like this. Like this, see, double stranded means both the sides will be there. Now, if you reduce the size and see it will look like chromatin, looks like a snake, it will look like a snake structure, it will look. Again, if you keep condensing, you will see like this. Then again, so this is how it is happening, okay? So it is going from DNA to nucleosomes to chromatin, from chromatin loops to your chromatin fibers, then to your chromosome, okay? This is how you see it under the microscope. So most of the time during the prophase, you will see like this. You will see one circle and you will see full, you will see like this, right? That is because here what you are seeing is your chromatin fibers. Chromatin fibers. This chromatin fibers, when you see it in your metaphase, you will be able to see as chromosomes, okay? This is how the structure is. Simple, easy, very easy, no? Yes, easy, okay. Now students, there are 2 types of chromatin. After this, maybe we can stop the class, okay? After this, maybe we can stop the class. 1 second. Yes, we can stop the class after this. Okay, so students, 2 types of chromatin is there. First one is euchromatin. Euchromatin, right? And heterochromatin. So euchromatin is the one, okay, here. Yes. Euchromatin is the one which will be loosely packed. This is the euchromatin which you see. I think I'll write it in that order only, otherwise you will get confused. So here we will write heterochromatin. Heterochromatin. This is heterochromatin, okay, densely packed. Densely packed. Because it is densely packed, it will be darkly stained. Is it understood? If something is kept very close to each other, I put a stain, what will happen? It will look dark for me, right? It will be darkly stained. Darkly stained, okay? Then students, one more thing is there, is it transcriptionally active or transcriptionally inactive? Your heterochromatins are transcriptionally inactive. Transcriptionally inactive. Okay, then you have euchromatin. Okay, euchromatin, first thing, it will be lightly stained. Okay, lightly stained. Second thing, it will be here, just look there, same thing, here it is loosely packed. And transcriptionally active. Ulta you write, okay? Vice versa. Simple. Okay, here it was densely packed, here it is lightly stained. So here you always, everything will be LL, there it will be DD. Okay, so you heterochromatin, you remember D, letter D, you remember euchromatin, you remember the letter L. That's all. Simple, dimple, easy. Okay, done? Done? Okay, that's all students. So next we have experiments. Experiments will start tomorrow. As of now, we will just revise what we did. I'll send you the fully written notes, okay? Do not worry. Yes. So we started with molecular basis of inheritance. We just understood what and all we are going to do. So today we did till bonding. We did nucleotide, genetic material. Here instead of experimental setup, we did packaging. Right, we did packaging, so nothing to worry. Now, in first we understood that Frederick Wisher is the person who found nuclein. By mistake, like he was doing some other experiment, he happened to find nuclein, which became the genetic material, right? And then Altman understood that nuclein has some kind of acidic nature in it, because of which he called it nucleic acid. Now, nucleic acid is of 2 types, DNA and RNA. DNA is called deoxyribonucleic acid, RNA is called ribonucleic acid, okay? Then we wrote the full form and we understood where it is synthesized, what it is, which it is the genetic material, which is not the genetic material. Then we understood some features about DNA, that it is a long polymer of deoxyribonucleic acid, and then length of the DNA depends on the number of nucleotides, and then nucleotide is what? 3 components. Okay, many nucleotides together forms the polynucleotide. All this we understood, and then we understood about the DNA or RNA also, that there is a presence of oxygen, because of which it is not very stable compared to your DNA. Okay, and then we understood some things that you have to buy hard. Okay, here students, I have put a quiz. It is nothing but sperm cells and egg cells are the only one in your body that is haploid. Yes or no? Haploid, right? So the value will be 3.3 into 10 to the power 9 base pairs. These are the only ones who are haploid. But if it is diploid, like a hair cell or a nail or something, they will be what? 6.6 into 10 to the power 9. That's all I have mentioned here. So just remember this. Your gametes will be haploid. So you should always write half the value. For other everything, it will be diploid. Okay. Now, then we drew the structure and understood the nucleotides, phosphate group, sugar, nitrogenous base, all this we understood. Then we understood purines and pyrimidine structure, where the ends are attached, then what is the binding, then how is the bonding, all this we understood, right? Okay, what is happening here, why is it 5' to 3', okay, why it is 5, then we understood beautifully the bonding, okay, 2 types of bond was there, we understood that also. And again, I showed an image. Then we understood how the discovery of double helical structure happened, who was the people behind this, and what did they find out of this. And that's how they found double helical structure. Watson, Crick, Franklin, and Wilkins, all these people who are helping in the double helical structure, out of which these 3 people got the recognition. Franklin did not get the recognition. Then we understood about nitrogenous bases. We understood about Chargaff rule. Okay, very, very important. We understood about Chargaff rule, and we understood what is right in Chargaff rule, what is wrong in Chargaff rule. We also solved one PYQ question. Okay, then we understood salient features of DNA. What is so special about DNA? Why is it the double helical structure? Right, this is how the structure is. Right, double helical structure. To be frank, students, we should always draw it like this. Should draw it in the same direction, but people don't draw it like this. People always draw it like a hair, braiding of the hair. Yes. Then again, we understood central dogma, we understood the multiplication, what is central dogma, and we understood how is the packaging of the DNA in the most beautiful manner. We understood the beads, we understood how the threads should go around each other, what are the histones which are there, how many of them will be there, which is the linker protein, what are the amino acids present, everything we understood about the packaging of the DNA. Yes or no? Did we miss anything? No, right? We understood the entire packaging of DNA, and we also understood how it is forming the chromosome, right? How it's forming the chromosome at the end. Then we also understood non-histone chromosomal protein, which will be used especially in your eukaryotes, okay, for higher binding. Okay, so it's nothing but, first starting it will be like this structure, right? Beads of string and all this, and then later it will form condensation will happen. That condensation will again happen to your chromosome. That's all. This is the structure. If you look, actually it will be like this. It will be like this. It will keep going like this, and then it will be histone proteins, like this. The structure will look like. Okay. Yes. Did I draw too much with the same color? Okay. Right, like this it will be. And like this at the end. Oh, what did I do? Yes, students, 1 second. Yes. So here we understood, what is happening here, from DNA to nucleosomes to chromatin, and what is happening here also we understood. And here I was trying to say, see, these structures will go like this, continue, and then finally it will form your chromosomes, right? All of you would have drawn your chromosomes, sister chromatids and all in your smaller classes. No, this is how it will form. Okay? Yes. Then we understood types of chromatin. Okay, types of chromatin. 2 types are there. Okay, types of chromatin we understood, hetero and euchromatin, and that's it. Okay, then we are doing experiment tomorrow. We will do Griffith's experiment tomorrow, or next I think I don't know, student, Thursday. Griffith's experiment we have to do. Okay, so that's all for today's class. I hope you really like the class. So let me know in the comments if you understood the class, And if you have anything to tell me, okay, you can let me know in the comments. I will see the comments. And thank you so much for joining. See you all. Bye. So if you want all the notes, please do join us on Vedanto NEET English channel. And if you're a student who is a NEET aspirant and who is finding difficult to find good content, then do join us. And if you understand English and if you feel comfortable in English, then please do join us on our Vedanto NEET English channel, and you will be able to get all the classes for free. Detailed lectures you'll be getting. Okay, clear? So thank you so much, students. Bye. Take care.